Hello, I am Andrea, an official SAT tutor with MMT Prep, and in this video I will be reviewing the answers for the third reading passage of the College Board's official practice test number 1. So this passage we can see instantly is a science passage. It has a little chart, and so it's very, very skimmable. I will do the skimming together first, and then we'll go through the answers. So we're going to start by reading the blurb. This passage is adapted from J.D. Watson and FHC Crick, Genetical Implications of the Structure of Deox, whatever that is, acid. Okay, so what I just did there is actually what you should be doing anytime you come across a really long word. You just want to register it kind of visually. It's almost like you're taking a little photograph, or it could just be like the big long D word, right? And you don't want to get caught up about how you don't know how to pronounce it or you don't know what it means. Try and define it for yourself as you keep going and just have that bookmark in your head, the long D word, okay? Do not get caught on the long, difficult words. This is super, super important for science readings like this, where you have like a weird word, and also for the history readings. Work around what you don't know, define it for yourself if you need it, but don't get tripped up on unimportant long words or pronunciation, other details, okay? We have to keep moving because we're answering questions. So I'm going to keep reading. 1953, by Nature Publishing Group, Watson and Crick deduced the structure of DNA. So we have an easier, much easier pronunciation right there. Using evidence from Rosalind Franklin and R.G. Gosling's X-ray crystallography diagrams of DNA and from Erwin Chargaff's data on the base composition of DNA. Okay, so we actually get a lot of information in this blurb. It's rare to get that much in a science blurb, but we're going to use that to our advantage, okay? So they deduce the structure of DNA using data from other people. So this is going to be about the structure of DNA, I'm going to assume, but I'm going to check. And so the first thing I'm going to need is my thesis. Usually the thesis is the last sentence of the first paragraph, but it can also be the first one. And so if I just glance at the first line, the chemical formula of DNA is now well established. Instantly I can see like, okay, that's going to be what they're talking about, probably. I can check that at the end they're still talking about it. Not exactly, but it's still related to DNA. I know this is all going to be about DNA, the structure of DNA. And so I'm just going to use that as a temporary thesis. Something about finding a thesis is like make your first best guess. If it's wrong, you can always revise it as you're reading to see the main point as it's moving through the piece. So for now, I'm just going to say it's about the chemical formula of DNA. And I am going to look at the last sentence of my paragraph from 10. Now I'm just looking paragraph by paragraph what's in each one. The monomer unit consisting of phosphate, sugar, and base is known as a nucleotide. Okay, so I'm going to assume this is just like basic definition of terms in this paragraph. It could be useful. I might want to come back and get it. But for now, I'm going to keep reading. So, second paragraph from line 12. The first feature of our structure, which is of biological interest, is that it consists not of one chain, but of two. Okay, so this paragraph is about 2 chains. This is where having a little bit of background information is helpful, but you can't use that background information to answer questions. It just means you'll understand more quickly what they're talking about. So now the paragraph was about the structure, the 2 chains. Now I'm going to look at the next paragraph from 20. The other biologically important feature is the manner in which the 2 chains are held together, which is by hydrogen bonds. Okay. So that paragraph's how their 2 chains are held together. Next paragraph, 31. We believe that the bases will be present almost entirely in their most probable forms. I'm not totally sure what this means. I'm going to keep reading. I can skip to the end. I'm just going to keep reading. It's not that long a paragraph. If this is true, the conditions for forming hydrogen bonds are more restrictive, and the only pairs of bases possible are... Okay, so the only possible pairs of bases. And then they list them. When you have learned about DNA, they do talk about that a lot. So if you're familiar with it, it makes this easier again. but they're only sets that these have to be paired with each other. Now I'm going to look at the next paragraph, 39. The phosphate sugar backbone of our model is completely regular, but any sequence of bases can fit into the structure. Okay, so it's a regular structure, but then there's any sequence. So it's not super hard to understand. I'm going to check in with the final sentence. Thus, one chain is, as it were, the complement of the other, and it is this feature which suggests how the DNA molecule might duplicate itself. Okay, so I am understanding what they're saying because I've learned about DNA, but that's a little bit of a difficult sentence. So if I don't totally get it, I can always skip back to the sentence before from 45. If the actual order of the bases on the one of the pairs of chains were given, one could write down the exact order of the bases on the other one because of the specific pairing. Okay, so the pairing on one chain defines the pairs on the other. I feel like I have a fair understanding of what this article is about. I know what is in each paragraph. And that second feature, knowing what's in each paragraph, is the most important part for the SAT for those detailed questions. So before I even look at the questions, I'm going to make sure that I am fully aware, it's totally concrete in my head, what's in each paragraph. I'm just going to go through paragraph by paragraph. First paragraph introduces our familiarity with DNA. Second paragraph is about the two-chain structure. Third paragraph from 20 is how the chains are held together with hydrogen bonds. Fourth paragraph from 31 is the possible pairs of bases. And then the final paragraph is that if you know one line of bases, you'll be able to find the other, and that's how they duplicate. Okay, you can come in any sequence, but that's how they duplicate. So I feel like I have a really clear understanding. I know where everything is in this article, even though I read very, very little. This kind of skimming is super helpful for science. It seems funny to read less, but it means that I have a clear understanding of the most important things, and I haven't gotten distracted by all these long, difficult words. Okay, so with that in mind, I'm going to start looking at my questions. 22, the author uses the word backbone in lines 3 and 39 to indicate that. Okay, so I'm going to go to those lines. So in line 3, the molecule is a very long chain, the backbone of which consists of a regular alternation of sugar and phosphate groups. Okay, so I'm going to say like the core of which, or the like central string of which, and then I'm going to look at my answer choices to see which one would match with that. This idea of like a core central string. Only very long chains of DNA can be taken from an organism with a spinal column. Definitely not actually about a spinal column. That's a classic trick answer. B, the main structure of a chain in a DNA molecule is composed of repeating units. Okay, possibly. C, a chain in a DNA molecule consists entirely of phosphate groups or of sugars. Entirely, this is an extreme word. I'm not going to want to choose that. D, nitrogenous bases form the main structural unit of DNA. This is simply false. So, the main structure of a chain in a DNA molecule is composed of repeating units. B, that is going to be my best answer, and it matches with their main claim about the structure of DNA. Okay, now I'm going to keep moving. 23. A student claims that nitrogenous bases pair randomly with one another. Which of the following statements in the passage contradicts the student's claim? Okay, so this is where we're going to find that the bases only have one possible pair. So I'm going to want to look in the paragraph that talks about that first, which is the paragraph from 31. I don't actually have a piece of evidence here, though. So I'm going to look at the closest one, D, and see if that would refute the claim that it can be paired with anything. So 27 to 29. One member of a pair must be purine and the other pyrimidine in order to bridge between the two chains. Okay? So this is saying that there's a definite, one has to be this, one has to be that. It cannot be a random pair. That seems to be a good piece of evidence. I can check the others, 23, 25. The bases are joined together in pairs, a single base forming a chain, one chain being hydrogen bonded to a single base from the other. This is saying that they form pairs. It doesn't say that they have to be a certain set, though. So it doesn't exactly disprove the student. The first two are background information. I can pretty much be certain that they're not going to be the right place. Okay, especially because I already have such a promising piece of evidence, 2729, that's going to dispute the student. I'm going to choose that and eliminate the ones from the introduction. 24, in the second paragraph, what do the authors claim to be a feature of biological interest? Okay, so this is just the topic sentence of that paragraph, and it's about it being of two chains. So this is where skimming is super helpful, because clearly the answer is going to be D. I didn't read that paragraph. I still haven't read that paragraph, but this is asking about the topic of that paragraph, and I am very, very clear in my mind of what that topic is. I haven't been distracted by other information. Okay, so let's keep going. 25, the author's main purpose of including the information about X-ray evidence and density is to... Okay, so I don't know where this is. I'm going to be looking for it. We have this great assistance that X-ray is very, very scannable, where you can kind of glance on the page and see it right away. And so indeed, I can find X-ray. And then I'm going to read that sentence and figure out what it's doing within my paragraph to be able to answer this question. However, the density taken with the X-ray evidence suggests very strongly that there are two. Okay, so it's providing evidence for the structure of having two chains. And so I'm going to look for an answer that is claiming that. So A, establish the DNA is the molecule that carries the genetic information. No, that's not what it's doing. It's talking about the structure of two chains. B, present an alternate hypothesis about the composition of a nucleotide. It's not alternate. It cannot be B. C, provides support for the author's claim about the number of chains in a molecule of DNA. That looks pretty perfect. I'm just going to check D, confirm the relationship between the density of DNA and the known chemical formula of DNA. It's not about the chemical formula. It's not going to be D. So, moving on, 26. Based on the passage, the author's statement, if a pair consisted of two purines, for example, there would not be room for it, implies that a pair... so this is where... the piece of evidence that we use for the student. It's right after that, and I'm going to say that it implies that somehow they would be too big because they wouldn't fit. I'm not sure, but I'm going to make that as my guess, and then I'm going to look through my options. Of purines would be larger than the space between a sugar and a phosphate group. It's not really, I'm not wanting between these two things. So that really doesn't make that much sense. B, of purines will be larger than a pair consisting of a purine and a pyrimidine. I'm not positive what a pyrimidine is, but I know from the sentence that I gave to the student that a pair must be a purine and the other a pyrimidine. Okay, so this is where you want to use those long vocab words just kind of as fillers and move them around the page. You don't necessarily need to understand them, you just need to track how they're being used, okay? So B looks like a promising answer. Two purines would be too big compared to a purine and a pyrimidine. I'm just going to check C and D. Of pyrimidines would be larger than a pair of purines. This is the opposite, and a wrong comparison. It can't be a pair of either. D, consisting of a purine and a pyrimidine, would be larger than a pair of pyrimidines. This, again, is an opposite answer. So I'm going to choose B. Two purines would be bigger than the purine and the pyrimidine, which is possible according to 27 to 30. So, moving on. 27, the author's use of the word exact, specific, and complement in lines 47 to 49, the final paragraph, function mainly to. Okay, so I want to make sure that I reread that section and have really clearly in my mind what those words are doing. If the actual order of the bases on one pair of the chains were given, one could write down the exact order of the bases on the other one because of the specific pairing, thus one chain, as it were, the complement of the other, and it is this feature which suggests how the DNA molecule might duplicate itself. Okay, so these are emphasizing that there's only one possibility and that that's how we are able to duplicate, the molecules are able to duplicate because of that singular possibility, and so it's very precise. So once I have that clear understanding in my mind, I'm going to look at my answers. A, confirm that the nucleotide sequences are known for most molecules of DNA. It's not saying that they're necessarily known. The pairs are known, so that's not going to work. B, counter the claim that the sequences of bases along a chain can occur in any order. No, they can occur in any order. That comes earlier in the paragraph. C, support the claim that the phosphate sugar backbone of the author's model is completely regular. It's not necessarily completely regular, and this isn't really supporting that claim, so it can't be C. D, emphasize how one chain of DNA may serve as a template to be copied during DNA replication. So that's exactly the answer I had in my mind, and that's what I was looking for. I'm going to choose D. So 28 and 29 are based on the table. So 28, based on the table and passage, which choice gives the correct percentage of the purines in yeast DNA? Okay, so I'm going to look at the table first, and I want to know everything that's in my table, what I'm looking at. So percentage of base in organisms' DNA, and then I have these different long vocab words and percentages, and then I have different organisms down the side. The question is asking about yeast, so I'm going to want to be looking at the yeast row, and then I need to figure out what a purine is, right? Because up here, my words aren't purines. So I'm going to go back, try and find the first time I see a purine, which is going to be in my first paragraph. 2 of the possible bases, adenine and guanine, are purines. Okay, so my 2 purines are going to be here. And then it's just going to be a matter of adding them together or looking at them both and looking for one that would match that. And so for 28, we can see clearly the answer is going to be C. Again, with this kind of chart-based problem, often it's easy to see an exact answer instantly, and we don't need to look through the alternates. There's only one possible right answer. 29. Do the data in the table support the author's proposed pairing of bases in DNA? Okay, so we would have to think about how that would be possible for them to do it, and then decide whether or not they would and why. Okay, so first thing is that we're going to need to know the author's proposed pairing. So I'm going to go back to the paragraph that was talking about only pairs of bases possible, and that is adenine with thymine and guanine with cytosine. So then if that were true, I would expect cytosine and guanine to be the same and adenine and thymine to be the same. So these would be a pair, and then these two would be a pair. And so I want to make sure that that looks like it's about right, and if they are looking that way, then it would support that evidence. And indeed, throughout the table, the 2 outer columns are closer as are the 2 inner columns. That does seem to be supporting it. So I'm going to go for a yes. So it's going to be between A and B. And then I have to think about why. So because for each given organism, the percentage of adenine is closest to the percentage of thymine, and the percentage of guanine is closest to the percentage of cytosine. Indeed, that's exactly what I was looking for. I can check B and make sure that it's wrong. But the moment that I look at it, because for each given organism, the percentage of adenine is closest to the percentage of guanine, I know that's wrong. Those are the 2 purines I used for my last question. Okay? So I can keep moving on. 30. According to the table, which of the following pairs of base percentages in sea urchin DNA provides evidence in support of the answer to the previous question? I'm going to want to look for sea urchin DNA. And then it's either going to be the 2 on the inside, remember these 2 that are going to match, or the 2 on the outside. So it could be 17.7 and 17.3, or it could be 32.8 and 32.1. So these ones are all a mix of the 2. It's not going to work. These 2 are the ones that are closest together. Indeed, I have 17.3 and 17.7, so the answer for 30 will be A. Moving on. 31. Based on the table, is the percentage of adenine in each organism's DNA the same, or does it vary? And which statement made by the authors is most consistent with that data? Okay, adenine definitely is not the same for all the organisms. So it's going to vary. And so it's going to be between C and D. And the place that they talked about the possibility of it varying would be at the end, where they're talking about the sequences, most likely. And so I'm going to look at those, or evidence from this paragraph first. And that's going to start lines 41 to 45. It follows that in a long molecule, many different permutations are possible, and therefore seems likely that the precise sequence of bases in the code which carries the genetical information. Okay, so many different permutations are possible. This does support the possibility of different amounts of adenine, and so I'm going to choose D. Okay, so that brings us to the end of this passage, and that means that we only have 2 more reading passages. One of those will be history, and then the final one will be another science one.